So what is this? I don't really know what lecture we're on. I guess
This is the
technically,
third lecture, I think.
what is that?
This is the third lecture, I
Yeah,
think.
but yeah, right. So we didn't, yeah, so this would be our third one. That makes sense. Kind of hard to keep track. This whole virtual stuff is quite interesting. And I gotta upload these. I keep forgetting to upload this stuff for you guys. So look, this is a math class, right? No one's expecting that you are you know much about neuroscience, or that that you have any in-depth knowledge of biology. So we're not going to assume that. And really, what's important to us in an introductory course is the math part of it. So I'm going to share some supplements for you guys if you want to dig deeper into them to learn more specifics of the biology aspect and the actual science, not the math. But if I can just break it down right now to sort of the very generic description of what we're going to do is, you imagine you have a cell, right? And you could think of that as a sphere if you want. And this cell has an electrical charge with it, and it has charge inside, it has charge outside. There's also chemicals around. There's chloride and potassium. And this is all very fluid. These chemicals are going in and out of the cell. The electricity's changing. And one of the things we think we can do is we've done it. We do experiments that show us, and we can sort of quantify or measure what we call the potential difference. And that is the difference of charge between inside and outside of the cell, is one way to describe it. And so here's what we're setting out to do in the most generic of ways. And if a neuroscientist was reading this, or watching this, they'd be like, this is not exact. Or if a biologist was saying, they'd be like, you're sort of, being vague. And the point is, we want to be vague. Right? That's fine. So what experiment teaches us is it teaches us that this potential difference. So let's put it this way. So when I'm going to put science, because when I say science, I guess I'm meaning like the biology and then the neuroscience, the chemistry, whatever you want to call it. So what they tell us is what we can study the potential difference. So this is a word some of you may be familiar with, or words I should say. Maybe you're
Yeah, we learn it in general physics too.
Yeah, you probably do, right. So think of this as the difference between the sort of what's inside the cell versus outside of the cell. And again, when I use the word cell, what I would ask you to think about is some sphere. So what we do is we note that this value changes in time. And we denote it. So the typical way of doing this, V, I guess, has its history with voltage, I suppose. We denote it VT. So this potential difference is V, and since it depends on time, we write it as a function of time. Is everybody okay with this? I mean, you guys, I hope at this point we understand. And since we have a fair understanding of electric circuits, a basic neuron model is to use so what we want to do is we want to describe V sub t or V of t. We want to be able to describe the voltage at any given time. Is to mimic an electric circuit. So some of you probably are familiar with these things from physics, maybe even in a differential equation, in your differential equations course, you probably did first and second order differential equations, and you did applications of electric circuits. In fact, I know you did. Well, of course, Abdullah, you didn't, because I don't think you took differential equations yet, but so you're sort of on the outside looking in with that.
So there's a RLC model that we learned in
Yeah,
general
so
physics
that's
too,
very
something like
similar
that.
to what we're doing here. Absolutely. So the
Okay.
resistor capacitor circuit. That's exactly right. Because these chemicals and this electricity, again, they're moving around and they have to go inside and outside of this cell through these things we call channels, for example. And some of them are better conductors than other, and some are resistors. And see, these words you might be familiar with physics. Look, let me put it to you this way. The more biology and physics you know, more the easier sort of understanding where these equations come from. But you don't need to know all that. I mean, again, we're mostly interested in the math here. That's what we're interested in. Now, as the semester progresses, we're going to try to get you, obviously, to learn more. But it's funny, when I look back, when I did this stuff in graduate school, when I was, for example, writing my thesis, I did all this math and all this math and more math. And then it wasn't until the very end that I actually went back and learned what all the interpretation of the math. So in my opinion, being that we're math students and not neuroscientists, or we're not biology students, in my opinion, the best way is to do the math and then sort of backtrack and show you what the math represents. Because that's sort of how my brain processed it. And so I'm going to do it that way for you guys. But your point is well taken, Jingzi. If you know anything about these basic RLC circuits, you can Google that, you can read about them. And it helps you to understand where this equation is going to come from that we write down. Okay, so does anybody have a question? Okay, so here's what we know through experiment. So experiment so experiment. So let's just say we have a we have a neuron, a cell. I don't know what you want to call it. I mean, I don't want to bog you down with the technicalities right now. So suppose we stimulate a neuron with current. So right now, imagine we have some nerve on a table in a laboratory, and we take some little device that we can stick into that nerve, and we can put electricity through it. We can put a current into it. So if we're not stimulating this sort of neuron, this cell, if we're not stimulating it, it has some resting potential. Its potential difference is some value. And like I alluded to in those homework problems, you know, a common sort of resting value might be minus 70 millivolts, for example. So if you read the history of this, one of the original experiments, they used a squid. So
1952?
Well, it was before that. In the 50s, you had Hodgkins and Huxley who had.
Yeah, that's the guy I was reading.
Right. So that's those guys came along a little after. I think they were in the 50s. But you can really trace. That was what their monumental paper was on, was this, what they call the squid axon. But you could trace this stuff back to the late 1800s, actually, where scientists, I want to say, I'm not positive, I want to say it was an Italian, who, what he was doing is he was staining like neural tissues. And he realized certain things, that neurons sort of had some common themes. So there's different types of neurons. Again, I don't want to get into all these details, but we're just, that's why I keep using these words in the colloquial way. But what's important for us to understand, and this is the mathematical problem at its finest, is the following. So if I draw sort of a coordinate system, let me move this down. And I imagine that the vertical axis is my voltage, my V, my potential difference, if you will. Well, let's not write voltage. Let's just write V. We don't want to use too many confusing words because we're not defining them yet. And of course, this is time. So what experiment tells us is, and I guess let me be a little more clear, because we're talking about look, we have to decide how we're gonna, what context, or excuse me, what convention we're gonna use for this potential difference. And typically we use it in the context where it's negatives. So I guess what I'm saying is you have to decide if the inside's larger than the outside, do you do positive or negative? It's just a convention, just like up is positive, down is negative. And so let me refix this graph, because the way we really want to think about it is we're going to use these negative numbers. So we might think of this as being like, you know, like this. And so what experiment shows us is the following. We have some sort of, say down here at like negative 70-ish. The voltage is hovering around here. And then at some point, which I'm going to label a different color actually, we start to provide a stimulus. And when we start stimulating it, what happens is, of course, the voltage begins to rise. And then at some other point, which we call, which, you know, we say that the cell has a threshold. And once it hits this threshold, the voltage shoots up and then shoots back down and then goes back to rest. So this is sort of what an experiment might look like. So imagine, if you will, this might be data from an experiment. Now, the first thing you might think about is, why are you using these little squiggly lines? Well, we could imagine if this was not an experiment, but in real life maybe, where there's a cell that's actively under change, it's the difference is not going to be, say, -70 all the time. It might fluctuate a little here and there. And then in the body, we imagine that instead of us sticking an electric device on it to stimulate it, you imagine these cells are connected to other cells, and then they're sending signals to each other through chemicals, and they're sending signals to each other through electricity. And when they send enough, or the right amount of signal, if it will, it starts to stimulate, and the potential difference shoots up. And what we call that shoot up is, we call it an action potential. So if I label some things, and I know I didn't define these words yet, but let's label this height right here. We call this the threshold. And then we call this what's called the action potential, or the firing. And so depending on how much physics you guys understand, you might think of words like repolarization or hyperpolarization and all this kind of stuff that you might sort of put with this, what we see, this data. So this is what we see. We do an experiment, we see this. And so the mathematician in us says, okay, what are the physical laws, and what kind of differential equation do they give us? Because the solution to that differential equation should be a curve that matches the data, at least somewhat. So let me state this again in words on the next page. Okay, so using physical laws, whoa, what's happening? My pencil is going ballistic. So using physical laws, our goal, if you will, is to construct an equation. In this case, it's going to be a differential equation, whose solution mimics the above graph. So if we draw it in a more as a continuous function type scenario, can we describe a differential equation whose graph looks like this? Oops, that was bad. Let me try that one more time. So we're looking for the function whose graph looks like that blue one, because that would be a good indicator of how the voltage behaves, or the potential difference behaves. So this is our first goal, is to sort of, can we find a function that satisfies this? Does anybody have a question or a sort of comment about this? So again, we are right now. I'm skipping all the biological details. I'm skipping all these physical details, like things about flux and things about, you know, these different things that potentials and the Nernst potential. And this is all stuff that I will give you supplements to read about and ask questions if you choose. But this is not the class for that. This is the class for the math part of it. And I will do my best to answer any biological or neuroscience questions as I can. And if I can't answer them, I'll put you in the right place to find them. Does anybody have a question? Okay, so let's write down this equation because it's a friend of ours from what we saw before. So one basic model is the ODE. So I'm going to explain what these constants mean in a second. This is not a T. This is the Greek letter tau. That's a T. So I'll explain what this stuff means in a second. So let me fix that. That's a C sub L. So I didn't tell you what any of these constants mean yet. So let's put this in a nice red box. So before we do anything, does anybody have a question about? Well, actually, you probably don't. You probably do have questions. So let me try to answer them before you ask them. So what is I of t? I of t is our stimulus. How are we stimulating it? Are we just zapping it with the same amount of electricity? You know, for example, are we giving it like a 10 amp or milliamp injection? Or are we going to do every second? Is it going to be, you know, is it going to be linearly inputted? Is it going to be inputs, you know, it's some pattern? How are we injecting our cell? Is it a constant injection? Does the injection vary? If it varies, then this stimulus depends on time. And that's why we write it as I of t. So this is the stimulus. So what is tau? Tau is important. Tau is what's called the time constant. So this is an analogy for this time constant would sort of be, those of you who had physics before, you may remember Hooke's law about springs. And we imagine that every spring has some sort of spring constant. that we know that certain springs are harder to compress than others, right? And so we imagine that neurons are not necessarily unique, but they might have different sort of behavior, and some have different time constants than others. So we've now, there's two things that we've answered that are in that equation that we didn't. And by the way, this is just a basic differential equation, one that you've seen before. You've seen this kind of equation in calculus 2 and in different. This is just a linear first order equation. You know what V is, that's the unknown. Now there's two other things, or a couple other letters I should say, that we haven't told you what they mean yet. So we didn't tell you what V sub CL is. We didn't tell you what G CL is. And then we didn't tell you what and what A is. Right, so those are three letters that appear in that equation that we haven't yet told you what they are. Now you might, depending on how much science you had, perhaps you know, chemistry or whatever, you might take a guess at what C sub L represents. You might take a guess at what area represents.
Well, I know V stands for voltage, right?
Well, right. So what about V sub CL? What do you think that stands for? What about G sub CL? Well, you know, so in you know, maybe you guys know what CL is from chemistry class, right? So the V is voltage, but what exactly is the V sub CL? So this is a little more.
So you mentioned, is it probably because of like a chemical stuff?
Yeah, that's right. It has to do with the chloride. So this is called, and then the fancy word for this is what's called the chloride Nernst potential. Now you might want to Google that if you want. But basically what it is we want the net flux of, say, the chloride to be 0. And so this is sort of when that happens. So another way you could do this is you can use potassium instead of chloride. So again, I don't want to go into too much of the details, but basically we're studying this cell by imagining the interplay between current and chloride and how the chloride passes in and out of the cell. So another way you can think of this, and it won't get you into too much trouble, I don't think at least, is this is the resting potential. And if you think about it, this makes sense. Suppose you ask what's happening when the voltage isn't changing. Look at the equation, look at the differential. If we want to know what's happening when the voltage isn't changing, so that means dV dt is 0. When the left-hand side is 0, that means there's no current either because we're not stimulating it. why can we think of that? Well, the only thing that survives is you'd have the equation 0 equals V sub CL minus V. So that means that that's the resting potential. Think about that. Ponder it. All right, so what about this G sub CL? What would that, or what might that represent? Well, remember, if you take a physics class, what does the letter G represent?
Gravity.
Well, it does. Okay. What other? Okay, so when you're in physics 2, probably, whenever you do electricity, you learn about voltage and current, and the conductance is also represented by G. What is G? G is 1 over the resistance.
Oh.
Right. You might, I'm not saying you remember that.
Yeah.
Trying to stir your imagination here, guys.
Okay.
All right. And so that's what I'm trying to do. I'm trying to stir your imagination. And so this is the conductance. I guess I got to change colors. And then A, we'll save some time, is just the surface area. Right? You would imagine the surface area plays depending on what is the size of our cell. If it's big, that will affect versus small. Does anybody have a question about this? So this is a very basic model of a neuron. And what we want you to imagine, though, is, again, we're not saying this is exactly how the brain works. That's super far from true. What we're saying is if you take a cell, like the giant squid axon, and if you want to know what that is, Google it, because that was one of the first experiments was done on. And you stimulate it with some outside current, I of t. This equation does a fairly good job of showing what happens, at least in the sense that it agrees with experimentation. You go to a laboratory, do an experiment, you collect data, I solve this equation under the given conditions, and it looks sort of like the data that you've studied. Okay, does anybody have a question about this? Comments?
I think we're good.
All right, so again, this is an equation that you guys are all capable of solving given, you know, it looks very frightening, but don't forget tau, the only, you know, in the simplest case, we have a stimulus of, say, of a very simple stimulus, then this is an easy equation to solve. Now, the only difference is if the stimulus is not constant, then it becomes a little more complicated to solve by hand, but you can definitely solve it by hand. Does anybody have a question? And you might say, because if you could write this equation in other ways, but the reason it's written this way is because if you actually would draw out sort of your fake electrical circuit, this is what you would arrive at if you followed the laws of all that stuff, Ohm's law and all that fun stuff.
So in this equation, we're actually treating GCL as a constant, right?
Yes.
And surface area is just another number, pretty much.
That's right. Well, you know the
Okay.
surface area. If we're assuming it's a sphere, then you know the surface area, right?
Yeah.
Yeah.
Okay.
Yeah. Does that make sense?
Mm-hmm.
Now, the interesting way of solving this, though, is when I of t is a piecewise function. So, for example, imagine a pulse where we go like on, off, on, off. And then that means that I of t is like a piecewise defined function. Or imagine it's like sort of a battery that charges and then dies down. So it's like maybe sinusoidal like this. So solving the equation in those instances is fascinating. What I'd like you to do is we're going to ask you to solve this with a bunch of different eyes and plot the graphs. Now you can do it by hand, but it's going to probably be easier for you to do it using, well, maybe we'll ask you to do it by hand too. Fun times. Fun times indeed. Okay, does anybody have a question about this? And then the next thing we'll do is we'll ask, okay, what about the more realistic thing is, what if instead of us giving it sort of the current injection, what if we try to be more realistic and imagine that our cell or our neuron is receiving a stimulus not from current injection by you in a laboratory, but from other cells. We call those synaptic inputs. So we'll talk more about that and how that changes the equation. Does anybody have a question? Comment? All right, well, then what I think we should do is I think that what I'm going to do is I'm going to give you your next assignment, which is going to be to study this equation under some different scenarios. And then we're going to ask you to do some stuff, and hopefully you'll have questions about it on Monday. Or if even sooner, you're more than welcome to email me and all that stuff. Does anybody have any questions, comments? All right, well then we'll officially end right now, and then like I said, I'll post some stuff for you to check out over the weekend, and you know, some supplemental stuff with all this. All right, so I'm going to stop recording, and Zayshin, I think you said you want to stick around for some questions, and then Abdullah, I'm going to email you as soon as I'm done with him, and then we'll schedule something up. All right.
Okay.
All right. I will talk to you guys on Monday, if not sooner.